Okay, and we are live. Assalamualaikum everyone. Hope you're doing well. So आज का बहुत exciting webinar series हम शुरू करने जा रहे हैं. So we're going to wait a little while ताकि more of you can join us in. So we're just going to wait for some time and then we're going to begin this amazing webinar series. So let me just tell you a bit about this webinar series जिसे हम इंतजार करते हैं इसके बारे में. So this is something that's very exciting for me because हमें बहुत इनक्रेडिबल क्या कहते हैं साइंटिस्ट से मिलने का मौका मिलेगा और वो भी मुस्लिम वुमेन इन साइंस से. सो दिस इज समथिंग दैट्स वेरी अमेजिंग. तो आई हैव हैड अ लॉट ऑफ फन प्रिपेयरिंग दिस एंड आई थिंक kudos to the entire team of Khwarzimi Science Society for bringing about this amazing webinar series. And जितना मज़ा हम सबको इसको अरेंज करने में आया, I'm sure आपको भी उतना ही मज़ा आएगा इस webinar series में participate करने का. So we're just going to wait a bit more. And then we're going to officially begin our webinar series. All right, so just a few more minutes and then we will start. मैं बहुत ज़्यादा excited हूँ मेरे से, तो मेरी excitement contain नहीं हो रही, मुझे हो रहा है कि मैं जल्दी से ये webinar शुरू करूँ because आज की हमारी guest is so special. She is honestly a wonder woman in science. So I just can't wait to introduce her. I mean, when I was preparing for this webinar, मुझे इतनी excitement हो रही थी उनके बारे में पढ़ते हुए, उनके बारे में जानते हुए, and I'm sure you guys are going to also enjoy it a lot. So just a few more minutes and then we will officially begin. Okay, so I think we should start because it's already 8:01. हम शुरू करते हैं. We have enough people to start off with. So assalamu alaikum everyone. My name is Nimra Khuram and welcome to this amazing new series by Khwarzimi Science Society called Muslim Women in Science and Technology Stories from Around the World. I am super excited to be a part of this series because it is truly a celebration and a tribute to the brilliant, talented Muslim women in STEM. So we will not only get to know about their incredible work, but we will also get a glimpse into their life, as in what are some of the struggles they had to face, their motivations, and who they are in general. So I'm super excited about this, and I hope you guys are ready for an awe-inspiring time. So our today's webinar is called the Era of Regenerative Medicine. So we will be basically exploring what exactly is regenerative medicine and why do we need to be excited about it. So without further ado, I would like to introduce my guest speaker for tonight. She is an extraordinary lady who is, get this, she is a cardiologist, a scientist, and an entrepreneur. She is currently an associate professor at the Icahn School of Medicine at Mount Sinai, New York. She has a very versatile academic background with a BS in biology, chemistry, thesis in physics, a minor in political science, and a medical degree from Harvard Medical School. She has been honored by numerous organizations and has been awarded the Sitara Imtiaz Medal by the government of Pakistan. She was a TED Med Innovation Scholar. She won the Clinician Scientist Award in the field of cardiac regeneration. So I present to you Dr. Hina Chaudhry.
Hello, assalamu alaikum. How are you, everybody? Can you hear
Assalamu alaikum.
me?
How are you doing? Yes,
I'm
I
okay.
can hear
Can you
you.
hear me?
Yes, we can hear you. Can you hear us?
Yes, I can hear you. I'm just checking because the last time I did this Zoom conference from Pakistan, nobody could hear me properly. So I wanted to make
Oh
sure
no.
everybody hears me.
Yes, we hear you loud and clear. Thank you so much for joining us. I'm just so excited to be in conversation with you tonight.
Thank you for
So,
inviting me. So I have a PowerPoint lecture prepared in case you want to see some highlights of that, or was this just going to be a chat session?
no, we can definitely go ahead. If you have a presentation, we can go ahead and share the screen. So, I think we should just jump into the webinar. And so, I would like to begin by first asking you to basically explain us what regenerative medicine is, why we need to be excited about it, and what is the amazing work that you've done, because you have done such a phenomenal work in that field.
Thank you.
So,
Well,
yeah,
so the the concept of regenerative biology is to be able to regenerate organs that undergo damage or disease, such as the heart or the brain or skin. You know, if a person gets burned or whatever, there are plastic surgeons working on skin regeneration. I work on the heart. And the heart is the hardest organ, along with the brain, to imagine even to regenerate because the heart is one of those organs where it's believed to be terminally differentiated, which means that once you're born, you're pretty much born with the number of cells you're going to have for the rest of your life. And then when an adult has a heart attack, the cells don't grow back. If, for instance, you damage the lining of your digestive tract, that will grow back because that grows back every 9 days. But the heart, if you have a heart attack, it remains as a scar for the rest of your life, basically. So that has been my goal, is to learn how to regenerate the heart. So as a postdoctoral fellow, first I needed to learn more about the genes that make heart cells divide in the embryonic heart. And once we uncovered what genes were responsible for that, we were able to use that knowledge to drive that type of fetal gene program in the adult heart to make them, to give them the ability to divide after a heart attack. And that way
all right.
we made cells divide and repopulate the damaged areas of the heart. So we've really been successful with our strategies for regenerating the heart. We were also searching for the kind of stem cell that could actually differentiate into a cardiac myocyte other than embryonic stem cells, which, as you know, can form any kind of organ. But obviously, there are great barriers to be able to obtain embryonic stem cells. You have to get them from the early developing embryo, and that's unethical for most people's religious beliefs. And it, it involves obtaining embryos, which is not considered ethical. So we were looking for a kind of cell that
Okay.
could mimic embryonic stem cells and form cardiomyocytes. And we did discover a type of cell that can be isolated from the term placenta. When a baby is born,
Yeah.
placentas are started in labor and delivery rooms throughout the world. So it's an almost limitless source. As long as human beings are being born, we can harvest their placentas and obtain these cells. This was a lot of work to figure out what cells in the placenta would actually work, and it was a fortunate. It was one of those discoveries that was just a spontaneous thing when I was doing rounds on my patients, and I had female patients with heart failure that occurs in
All
pregnancy
right.
called cardiomyopathy. Half these women recover completely normally, and it's the highest rate of recovery we see in any form of heart failure. So we started thinking, is there a fetal component to this recovery? And we thought maybe fetal cells were traversing the placenta, getting into the mother's circulation, and getting to the heart, and thereby regenerating the heart. And that's exactly what we found, because we modeled this process in mouse models first. And so
All
we
right.
created, a mouse model in which the babies in the mother's womb were labeled green. And then we caused an experimental injury in the mother's heart, and we found green cells. We could track them
Oh,
going to the
traveling. All
mother's
right.
You know, so then of those green cells, we were able to, find stem cell markers on these green cells and determine what cell type was playing a predominant role. And once we found a marker that was very prevalent on these cells, then we made another mouse model in which we labeled just that cell type. So you can put genetic tags through genetic engineering approaches on certain type of cells. So
Okay.
when the cell marker we found was a CDX2 cell, and we had put a label on the CDX2 cells, and then we studied those green cells because they were all CDX2. So we could isolate them from mouse placentas, put them in petri dishes, and they would form literally beating cardiomyocytes. I have examples to show you in my
All
PowerPoint.
right.
And um,
Yeah.
and then we also could give them, get this, to male mice that had
Okay.
undergone a heart attack. And we would take them from the female placentas and we would inject them in the tail vein of the male mouse, and they went right to the injured part of the heart and repaired it. They have this
Oh
ability
wow.
to hold
That's
the site of
fascinating.
injury, which most stem cells don't. But they are sensing injury from the circulation. They go to the place of injury like a heat-seeking missile.
All
So this
right.
is a really exciting discovery, and now my laboratory just got awarded a very large NIH grant to study these cells in human because we're finding them in the human placenta as well. And we just did the experiment where we isolated them from human placentas, and we were trying to see in cell culture whether they are able to migrate towards this gradient of this protein that is released by the injured heart, and they also migrate. So we are replicating the results we
All
obtained in
right.
mice in the human system. So we're hopeful that we can develop a stem cell therapy for human beings using these
All
cells. And
right.
they contain all of the genes that are associated with the stem properties of embryonic stem cells. So we're very hopeful that we can use them for other applications other than heart disease, say for the brain or for the liver or the lung. We are actually have a project in which we're studying them for lung regeneration in the lung injury associated with COVID patients. So we are trying
Oh
to see
wow. Okay.
as well. And if I can share the screen.
Yeah. I think you would have an option to share screen coming on your laptop screen. You just have to
Yeah,
click that and we'll be able to share.
okay.
Yeah. Let me put that on the screen.
Do you see it?
Yeah. Yeah. I think everyone can see this. I'm just gonna remove myself from this so that everyone else can enjoy this. Okay.
I
All
don't
right.
see you now. I only see the screen.
Yeah, it's okay. I think everyone else can also see the screen now, and they can hear our voices.
Okay. So what you see here is an actual heart cell, and this is under the microscope with antibodies that are delineating a dividing nucleus because we're the first ones to figure out how to make mammalian cardiomyocytes divide, and that's the basis for any regenerative therapy. You have to be able to induce cell proliferation. So this picture is actually from the cover of research that we published back in 2007, where we, for the first time, described cells undergoing mitosis and able to repair the heart through the strategy of using this fetal gene that I had talked about, Cyclin A2, that I have characterized in the heart. Now, the next slide, and the next slide is basically a movie, a representative movie of what we're trying to achieve, that we'll be administering either the gene therapy or the stem cells through a catheter in the femoral artery, threaded through the aorta up into the heart and injected into the coronary tree. And you see these golf ball sized particles. They are the actual carrier of this vehicle in green that we are releasing. and it's causing the cardiomyocytes in the damaged area to divide and repopulate this scar area. So that's just sort of a digital movie depicting the strategies that we're talking about. Like I said, we're talking about two kinds of strategies. One is making the native cells of the heart divide, and that's based on lower organisms that are not part of the mammalian species. The mammals cannot have their heart cells divide, but there are lower species that can have their heart cells divide. For instance, this is the North American red-spotted newt, and it also can have its, if you take the heart of the newt out and you try to, you put it in a petri dish in cell culture here, these cells will re-enter the cell division cycle, re-aggregate to form a beating syncytium. This is not normally possible in the mammalian heart. Mammals include, as you know, all this sort of animals with hair, starting with, mice are mammals, we're mammals, and so things that work in mice often also work in humans, so we also always start in mice. Now, another animal that can regenerate its heart is the zebrafish. It's known that if you damage the heart, the apex, it grows back over a period of 60 days. None of these processes, they were not possible in any mammalian species before, and therefore obviously not in humans. So that's why I decided in my postdoctoral fellowship to study the cell cycle. And here it's cyclin A2, and I found it to be the critical regulator of mitosis in cardiomyocytes. And that's the basis of a gene therapy that I have devised now that I'm working with the US FDA to gain permission to test this in human clinical trials. Now, before you can go to the FDA in the United States and ask to test things in humans, you have to test them in a large animal where the heart structure and anatomy closely mimics the human. And so we did this testing in pigs because the heart and physiology and the anatomy of the pig is very, very similar, just about identical to the human heart. And we used 4 different state-of-the-art imaging modalities. Here is shown left ventricular angiography at the bottom, 2-dimensional echocardiography, 3-dimensional echo, and MRI to study the heart function in these pigs, which this is depicting the study design. We had at time 0 performed the imaging studies to assess the function of the heart, then created the myocardial infarction. 1 week later, we repeated the imaging studies to make sure that the MI creation was successful and that we had caused an infarction. Then we injected the virus into the heart. Then we waited another 6 weeks, repeated all the imaging studies, sacrificed the pigs, and performed histologic studies. So the bottom line was, if you look at this graph at the bottom, the ejection fraction, or the measure of the function of the heart, was significantly increased in the pigs that had received the cyclin A2 gene therapy. The control group received the same viral vector, but the viral vector was missing the cyclin A2. So we wanted all the other conditions to be the same, and their ejection fraction was going down. And this is just an example. It's probably too technical, but it's an example of how we studied the process of mitosis inside the cardiomyocytes. This brightly lit up nucleus is undergoing cell division. Excuse me. And the green staining is a marker for the cardiomyocyte structure, the actual heart muscle cells. This is a 3D analysis where we can see, excuse me, that the nucleus is embedded in the cardiomyocytes. There were a number of other studies that are just summarized here that we performed to show that we had successful regeneration. Next is a slide from a human male who's 55 years old. We isolated his heart muscle cells and used the cyclin A2 gene therapy. As you can see here, oops, I don't know what happened there. Let me get that back. I pressed the wrong button.
Yeah, it's okay. It's all right.
So getting to the human, this is a heart muscle cell from a 55-year-old man, and we've treated this cell culture dish with Cyclin A2, and over a 70-hour imaging period, it undergoes up to 3 rounds of cell division. Here's the first one. Here are the two daughter cells. And then at 65 hours, it's starting to undergo another division, and you see at 70 hours, these are the daughter cells resulting from the second division, and then one of the daughter cells is starting yet a third cycle of division. We can only image these cells for 70 hours, otherwise they'll die. These are very tricky research techniques that we actually invented in my laboratory, because the red fluorescence that you're seeing in the live cardiomyocyte is a fusion protein we made of a red fluorophore that is inserted into the sarcomere, the muscle structure of the cardiomyocyte itself. So this has previously been impossible to study because no lab in the world has succeeded in culturing human adult cardiomyocytes, and we had to develop those techniques ourselves. So here's the movie which depicts that cardiomyocyte, and this has never been seen before. These are results we're just getting ready to present at the American Heart Association meeting this year. You see the cell is undergoing division. Nobody has ever seen this. Is from a 55-year-old man. Nobody thought this was possible. This is only possible with the use of the Cyclin A2 gene therapy that we've developed. Next, I'm going to go on to our stem cell approach. As I was describing to you, we know that stem cells from the baby cross over into the mother and get into different organs and into the mother's circulation. That was known for decades, but nobody knew what the function of those cells were that came from the baby and crossed into the mother. And we knew that from our mouse studies, we knew that in the mouse, we could see blood cells, epithelial cells, hepatic cells, neuronal cells. In fact, a number of people had shown that cells that could be tagged in the mouse babies and in the mother's body, they would develop into these cells. We were the first to show that they could, develop into actual cardiomyocytes. And so this was our contribution, obviously, with the heart. And we had taken the green cells out in the mouse, and we looked for stem cell markers. And here is where I was telling you there was a predominant marker here by that's called CDX2. And it's a gene that was never previously thought to be involved in development of any organs. CDX2 is found in this layer of the blastocyst, the outer cell layer, that implants into the uterine endometrium shortly after conception. When cell division happens in the fertilized egg, this inner cell mass develops, and this gives rise to embryonic stem cells. There's an outer layer called the trophectoderm, and this is the CDX2 is a marker for the trophectoderm. And I think I always had wondered for many years why it was that everybody was focusing on the ES cells here labeled in yellow that were coming from the inner cell mass, and those unique properties of the outer layer that is able to implant into the uterine wall and give rise to placenta, that they must have very unique properties, and why is nobody really focusing on regenerative properties of these cells? So that's the cell types that we are talking about. And we found, like I said, we engineered a mouse. This is very technical. We basically engineered a mouse in which the green fluorescent tag was labeling all CDX2 cells. After we engineered this mouse, and we took the green signal was in the nucleus of those cells, and we cultured those cells in vitro in a petri dish, and they give rise to this beautiful cardiomyocyte, which you're seeing this red staining, the railroad pattern, is the hallmark of what a cardiac muscle cell looks like. So this nucleus, which is also fluorescing green, is clearly within this cell, which is clearly a cardiomyocyte. Therefore, we knew that these CDX2 cells were able to differentiate into these beautiful striated cardiomyocytes. And then the movie depicting the beating is right here. See, this is the green nucleus, and the cell that contains it is beating. You can see that right here. Oops. I think it played, the beating played for a little while. Now I'm having a hard time getting it to play. Okay, there you see the whole, like, muscle. This is a beating cell, and that's a green nucleus. So we knew these cells were functional cardiomyocytes. I'm going to skip over this slide. We found that these cells are able to evade the host immune surveillance system, and these slides basically just go over immune markers that we studied. And this is a protein signaling network that we delineated in these cells, and that's how we found how they are able to home to the site of injury. And we actually were able to block that homing process and showed that when we block the homing process, the cells do not migrate to the injured organ. And that's how we had found out actually the mechanism of how they go to the site of injury. And this is just more of those migration studies, which, as I said, we've repeated these now in the human cells. So far, we know the CDX2 lineage cells represent a unique multipotent cell type in the placenta, and they form spontaneously beating cardiomyocytes in vitro, and that they possess an immunologically naive gene signature, meaning that they can be in another host and they will not be killed off by the host immune system. Now, this is the coolest experiment in that study, in which we took the male mice and we caused infarction in the male mice. We took the cells from the female placentas, we gave them in the tail vein of the mouse, and we did MRIs several months, 1 and 3 months after giving the cells, and we found that they even repaired the male heart, that they homed right to the site of injury. This is the MRI ejection fraction of the CDX2 group. This is pre-treatment, this is post-treatment, the gray bar. So we saw a significant recovery of cardiac function. And this is also ejection fraction. This is at the 3-month time point. We see a significant increase in the cells in the mice that were treated with the CDX2 cells compared to controls. And these are MRIs depicting that. I think that one of my students who made this forgot to loop it so it doesn't play continuously. But here is a mouse that was treated with CDX2 cells, and the other one is the control. And so you can see that heart is dilated and doesn't contract as well as the one that was treated with the CDX2 cells. So this is more histologic studies just showing that the cells with the green nuclei, which come from CDX2, are actually differentiating into cardiomyocytes inside the heart, because this is a section taken from inside the treated heart, and this is just a sort of a zoomed in picture of that. This is just scientific proof that that the cells are doing what we say they're doing. Now our next goal is to create an ethically compliant and multipotent cell source for allogeneic cell therapy. Allogeneic means that you can take it from one individual and give it to another individual. And we're hopeful that we can do that because of the immune privilege these cells enjoy. So we are going to do that for cardiac and other types, as I mentioned, other types of organ repair as well. And we've been able to, this is a picture of the human placenta. And as I said, we've been able to find them and isolate them from inside the human placenta. And this is what we're working on right now. We're looking at CDX2 expression from these patients. We've done the sequencing. We have found indeed that those cells have the right sequences. This is just showing that they express the right proteins on the RNA sequences. And this is basically another experiment which we look at protein expression of CDX2, and we're finding it within these chorionic cells from the human placenta. So now we've developed a strategy to select live CDX2 cells from human term placenta. We're using a virus that expresses a red fluorophore to select them. And so where you see red, that means that we are able to select these CDX2 cells. And now this is our plan. We will be taking them from the human placenta. We will be administering them intravenously. This is future. This is not right now. To human hearts, and we expect that they will home to the injured zone, and they will not traffic to non-injured organs, and we will in this way be able to repair the heart. So we've worked out two different strategies. The first one was the gene therapy I talked about. This is a cell therapy, a stem cell therapy strategy. We've received a lot of press. This was a cover story from New Scientist magazine a few years ago, um, talking about this work, how your mother cells are in your body and your cells get into your mother's body, and what that all means for human biology. And this is sort of just the acknowledgments for the people who have worked my entire lab that has been working very hard on these studies. We have collaborations with Cedars-Sinai Medical Center. They help us with all the protein analysis that we are doing, and we've received quite a bit of funding, New York State, Stem Cell Board, the NIH, the American Heart Association, as well as private venture capital. And this is a current picture of my current group, and obviously because of COVID, we have to work with masks on. So that's basically the highlights of my work, and I'm happy to take any questions that anybody has.
Yeah, so that's just brilliant. So we do have a question. I'm just going to display it on the screen, and I'm going to read it out loud as well. So this is a question from Dr. Amir Faisal, who's asking us what are these CDX2 expressing cells? Are these trophoblast stem cells from the, I'm pronouncing it right, I'm sorry, I can't pronounce this word properly.
They're
Are
trophoblast
you,
stem cells.
yeah.
Yes, they're trophoblast stem cells. They're embryologic remnants actually that we're able to find in the term placenta. There's just a small number of them, but they're still there. And it was surprising to us because the scientists who had really studied CDX2 and its involvement in placenta formation kept telling us that we would not find them at end gestation in the term placenta, but we're able to find them.
All right, so we do have another question, and I'm going to display it again. This is by Dr. Mohammed Mustafa, and he's asking if you're using placental CDX2 directly to repair the heart or it requires another treatment.
Are we using it directly, or does it require some treatment? Oh, in so far we've tested them in mice, and we use them directly without any pre-treatment whatsoever. So we're hoping that we can do the same thing for human, if the human turns out to have the same properties as the mice do.
Right, so how long do you think that this could actually take before it actually comes into clinical trial and we start testing it out on humans?
So with
This
our
is
gene
amazing.
therapy, for our gene therapy, we're hoping that in a year we'll be testing in humans. For the CDX2 cells, it will take a little bit longer. I think what we're going to try to do is test it in monkeys in the primate model. So it might be a good, I think, maybe three, maybe four. If we're lucky, maybe 4 years until we can test it in humans. So.
Yeah, all right, that's just brilliant. I'm just really in awe right now because this all seems like science fiction coming into reality because nobody would have imagined this happening before, and this is such phenomenal work. Thank you so much for sharing that. So now I'm just going to go ahead and move to the next section of the webinar, and this is what I like to call a heart to heart. And this is where we will be talking about all the motivations and the struggles you had to face to get to where you are today. So are you ready for that segment?
Yeah, sure. It's not been easy. None of this has been easy. I'll tell you that. It's been very challenging. Yeah.
All right. So the very first question is: so what was the inspiration or drive that made you like a moment where I'm going to become a cardiologist, and then it transitioned into becoming a scientist as well, and then an entrepreneur. So what was that moment? What was the inspiration?
So I was, we South Asians live with a lot of heart disease. Everybody, everywhere you look, has heart disease, and the same was true in my family. My mother, who is now like 80 plus, has 5 stents in her heart. Thank God she's never had a heart attack, and I aim to keep it that way. But she has significant coronary disease. My late father, who passed away 10 years ago, when I was just a 7-year-old child, he had a pretty serious heart attack, and he passed out in our driveway. And because I was only 7, it really affected me. I was the eldest child, and we had to grow up with always being quiet in the house so that Daddy wouldn't have another heart attack. And this was sort of, my mother used it to keep us, well behaved and quiet in the house to protect our dad. And so it had a big effect on me that this is such a serious thing. And I was so grateful and amazed that a year later he underwent quadruple bypass surgery, and he did great and he lived. And at that time, quadruple bypass surgery was no joke. The heart lung machine had not been around for that long, and there was no guarantee. Mortality was pretty high on the heart lung machine. So we weren't sure that he was going to survive the surgery. And when he did, and he lived a very full life and had 2 careers side by side, I was just so grateful to his doctors that I felt like, I want to study this disease. And it's great that he was helped, but you heard so many other stories and other children's fathers not making it. And so I really wanted to understand, how does heart disease happen, and what are new ways that we can fix it without having to go through surgery and all the stress of surgery. And that's why I was interested in studying how to regenerate the heart after a heart attack. So that, even my father's heart, when I was 7 years old and he had an inferior MI, if you fast track to 30 years later, and I was a fully established cardiologist by then, and my father was at the time still alive, and I performed an echocardiogram, an ultrasound of his heart, where he'd had the heart attack in the inferior wall 30 years before, there was still a scar there. So you could see the scar. It doesn't regenerate. He was 47 at the time he had the heart attack. And 30 years later, you could still see the scar. So there was absolutely no regeneration. And that is really what motivated me to study this. And then when you come up with a new therapy and you want to test it in humans, you have to go through a commercialization route. There's no way that you can go from bench research in an academic lab right into humans. You have to go through this commercialization pathway, and that's what prompted me to form a biotechnology company and do this through private capital.
Right. So yeah, wow, that's really inspiring. And I guess that something like this, when happens in your life, that sort of shapes the thing that you want to pursue. So the next question is, being a woman in science is not easy at all. So what were some of the struggles that you had to face?
There are a lot of sacrifices when you come from Pakistani culture and you come from, have traditional parents. There are a lot of sacrifices. We work day and night. This is not easy. Just the night before last night, half my lab was here until 10:30 at night. I'm fortunately not at the stage where I typically stay that late anymore, but my lab members who are actually doing the research, they're here at crazy times. So when I was younger and I was actually doing the bench research myself, I was also in the lab all night long sometimes. So there's a lot of sacrifice. It's not easy. And then on top of that, the hardest thing for me has been that I was a young trainee and a young faculty member right after 9/11. And even though I've been raised my entire life in this country, I'm basically more American than Pakistani. I speak with an American accent. I had to deal with all the Islamophobia because obviously my name is Muslim, and I feel like, especially in New York, I've dealt with a ton of Islamophobia in the academic world. And I think it's getting better. You just see so many more young people of Pakistani background, of other Muslim country, backgrounds that are doing really well in academia. But in my time when I was trying to establish myself, it was incredibly difficult. And I feel like so many obstacles have been thrown in my way. First of all, just being a woman. You have a great idea and it actually works. The male colleagues, I don't know, a lot of my female colleagues who were professors in Muslim countries said that it was actually not that bad. I have friends who've been professors in say Dubai or Turkey, and they're saying that we have women in the United States have it actually worse. And you can see that. I mean, we have a vice presidential candidate who's a woman, but we've never had a woman president in the United States. So to be honest with you, attitudes against women in many ways I think are even worse in the United States. So just if you're a bright woman of any background with great ideas, it's always a problem in academics, you know. So it's been actually a huge problem. So you just have to have a very thick skin, and you have to learn how to, you know, deal with nasty people.
Yes. I truly understand that, and I understand. I think a lot of us listening understand when it comes to belonging to a Pakistani background, a traditional Pakistani background. All right, so I'm going to take a question that is from Dr. Saad Anwar Siddiqui, who is the president of Khwarzimi Science Society. He is basically asking you that what is your message to the youth of Pakistan working in interdisciplinary fields of life sciences?
I would say, you know, I don't know the state of secondary education in Pakistan. I just think that it's hard to have this sort of exposure to research in most countries other than the United States. Even people in Europe, they want to come here for their postdoctoral training. So once you get your basic education, say you get your PhD or your MBBS, I encourage them to try to do postdoctoral training in the United States, or possibly England. Is pretty good. It's not as good as here. To really dive into what they're doing, because life is short. When you're young, for instance, the young people in my lab are, like I said the other night, they were working till 10:30 at night. You only have that energy when you're young, and you have to make your mark. And your great ideas are going to come when you're young, and that's going to be the only time to really develop them. So in your 20s and early 30s, I would just say really focus on your work and making yourself the very best at what you do.
So, coming back to the struggles that you talked about, so how did you actually overcome those struggles to be where you are right now? So, what are
I've
the
had
do's and the
very good
don'ts that we need to
lawyers.
know?
I've had very good lawyers. Let's put it that way. I've paid a lot of money to excellent attorneys. Sometimes that's what you have to resort. You know, on my father's side, we've got that Afghan blood, that Pathan blood. We're very tough, so we know how to fight. You have to. You cannot be a pushover and be a woman in science. You cannot be that way. You really have to. When the need arises, I go to lawyers.
Also, we would like to know that have you had a collaboration with any research group in Pakistan?
I've always wanted to, but I haven't. I think that a few years ago, somebody had mentioned I should maybe have something joint with LUMS. I would be very interested. I just haven't had the right connections to set that up. But I would love to. You know, I still have relatives in Pakistan. It would be wonderful to visit. I had given a visiting lecture at the Armed Forces Institute of Cardiology in Rawalpindi back in 2005. That's the last time I gave an actual in-person visiting lecture.
Okay, so that means we need to call you and start collaborating with you as soon as possible. So there is a possibility we're going to get Dr. Ina come to Pakistan soon.
I would, you know what? One thing I would love to do is help set up a research institution in Pakistan where there will be an exchange of people who could come back and forth and exchange ideas and train and things like that, where I could go and teach them something, or they could come here and do some training. I mean, I would be very interested. And I think that up till now, I've been so busy and getting these concepts, published and established, and we're obviously there's still a lot of work that remains to translate them into human therapy. It's very busy work. I won't, so I had wanted to be at a point where at least I've tested something in clinical trials, and I would have more time to help set up something in Pakistan as well.
So I really can't wait for this to actually happen, and I hope that it happens soon. So one question that I've been meaning to ask is that you have a very versatile academic background. So I wanted to know, how does a thesis in physics and a minor in political science fit into all of this?
I get bored very easily. You know, I wanted to study everything. I'm very interested in political science. I think the physics part was that I was working on a project on laser angioplasty. So I was studying the interactions of laser light and vascular smooth muscle cells. And therefore, my thesis happened to be in a laser lab, but it was very interdisciplinary. And that's the beauty of the United States. You can do so much interdisciplinary work here. I have, protein experts in Los Angeles, and I'm in New York, and we're working with them. I have scientists that understand the details of gene regulation downtown at Rockefeller, and I can go and pick their brains on that. So you have to collaborate. You have to reach out to people who have expertise in areas you don't have. We're now doing analysis of deep sequencing, and there's big data sets. So we need the experts in bioinformatics, for instance. So the bioinformatics people that are helping us, they're sitting in Los Angeles. Some are sitting in New Jersey. So it's this interdisciplinary nature that makes big discoveries and makes science move forward. You can't do it in isolation. You can't do it by yourself. And you just can't do it in one laboratory either. So you have to reach out and collaborate to people with other kinds of expertise.
All right. All right. So, what is a golden rule that, or some golden advice that you would give to girls and women who are trying to pursue a career in STEM? So, what is the golden advice that you think they should follow for sure?
Listen to your inner voice. That's the bottom line. You have to listen to your inner, don't listen to anybody else. You have to listen to what your own inner voice. And I think that the person who made me really was my father. I mean, he gave me all the freedom in the world. He gave me the wings to fly. And in our culture, to be honest with you, I'm finding the mothers hold their daughters back more. So don't ever listen to your mother. you know, I never listened to my mother.
I'm not sure how this is going to go with people, especially mothers who are watching this. Just going to be like, okay, no, you didn't hear this at
There
all.
are mothers who are different, but the majority of Pakistani mothers, they try to hold their daughters back in my experience. My father was the one, he sent me everywhere, he gave me every freedom, and that's the way it should be. And if the mother's like that, fine, listen to her. But if your mother's like my mother, who is probably gonna see maybe a taped version of this, I would just say don't listen to them. So
Okay, yeah, that's good. All right. So now we're going to move on to three silly questions that I'm going to ask you, and this is just to know your casual, non-scientific self. All right. So are you ready for those three questions?
sure.
So the very first question is, what is the weirdest food combination that you've tried?
What is the what?
What is the weirdest food combination that you've tried?
Oh, boy. I'm pretty adventurous when it comes to eating. So, food combination? Um,
Let me give you an example. I used to see my grandfather do this. He used to mix ketchup and like gravy together, like a shorba with ketchup, and it was really weird for me. But he really used to like it. So have you experimented something like that?
I would say nothing that weird. I just like, through my academic travels when I go give lectures abroad to try local cuisines. Obviously, if there's nothing, in them that is not allowed according to, Muslim belief, like no pork, no alcohol. But other than that, I'm always very interested in trying the cuisine of different cultures. But I don't find it weird. I just find it like an adventure. I guess I had more difficulty. I was in Sweden for my first invited talk in Europe, and the funny thing is that there was only one thing I liked about Swedish food, and that was their shrimp. the shrimp sandwiches. Otherwise, I didn't. It was too bland for me. So I used to go find, Indian or Chinese restaurants in Stockholm to eat. But usually I like trying the native food wherever I am. But I don't really consider it weird. But sometimes it's different from what you're used to, right? So.
All right. So the next question number 2 is, what extreme sport would you like to try? Like, for
Well,
example, would you like to bungee jump?
I actually do extreme sports. I'm a skier. I'm an avid skier. I do downhill skiing. I don't do all the dangerous slopes that I used to. So I've done the sort of what the Black Diamond slopes in on most of the Rocky Mountains. And as I'm getting older, I'm getting more careful because I have more responsibilities. But so I am an extreme sport enthusiast, and I've always enjoyed horseback riding. And I've actually been on a horse where it's reared up its front legs like Hi-Ho Silver, and I didn't fall off. So I do. I would like to. I do have one interest. There's a friend who is a software entrepreneur outside of Lahore, and he and his family have invited me that anytime I'm in Pakistan, I should come and visit their farm, and his children are going to teach me how to play polo. So that's something I'm very interested in trying. I've never played polo, but I've been on a horse. And so I would like to come to Pakistan and learn how to play polo, because what I tell all my Western colleagues, polo originated in Pakistan. The British learned it from the Pakistanis, not vice versa.
Yes, we should definitely have you over, and I hope that that happens quick and very soon as well. Okay, so the last question is, what is your dream vacation destination?
I love visiting Turkey. I've been there many times, and I hope to go many more times. But I know I've been to Istanbul because some of my best friends live in Istanbul, so I go and stay there in Istanbul with them. But I've not seen the so-called Turkish Riviera, the areas around Marmaris, Bodrum, Antalya. I'd like to go and vacation in those regions right on the Mediterranean, and that are about a few hours south of Istanbul. You know.
All right, that's lovely. Thank you so much. I mean, this really helps us to, humanize scientists, because we never think that, okay, they might also be thinking about these things and
Oh, one
all
more
such
thing. I want to
interesting
ski.
stuff.
I want to ski in Gilgit Baltistan. That's what I want to do. So anybody
Oh, all
listening,
right.
if you can help arrange that.
Yeah, so if anybody is listening, please arrange a trip for Dr. Hina in Gilgit so that she can come and ski for us. And so let us know about the adventures, though, because this is really interesting for me to know that a scientist is interested in extreme sports. That is something new for me.
Yeah.
All right, so thank you so much for these. So now what I'm going to do is we're going to take some more questions from the audience. So first of all, we do have one related to your research work, and I'm going to display it on the screen right now. So it's again by Dr. Amir Faisal, and he's asking if where is the wait? It's not coming. Are you able to see it? Apparently, it's not
Yeah,
coming
I
on my
can see
screen.
it. He's saying that cyclin A is involved in the S phase of the cell cycle. How does overexpression of cyclin A2 affect G1S transition? Actually, that's a little bit partially not true. Cyclin A2 is the only cell cycle regulator that is regulating both the G1S transition as well as the G2M transition. So it's the only cell cycle cyclin that is critical for both of those transitions, and that's why we have such a potent effect of making the cardiomyocytes divide.
All right. So then another question is that, have you ever visited Lahore?
Oh yes, many times. I just haven't been there in a long time, and now it's the whole COVID thing. But yes, I've been there. I have a lot of relatives in Lahore.
All right. So what is one of your favorite places in Lahore?
The Badshai Mosque.
Ah, nice.
And of course,
Right.
and of
So
course, and
yeah.
Anarkali Bazaar.
So yeah. Yeah. So that means you haven't been to Pakistan a very long time. So you need to have like a short visit soon. And we at Khwarzimi Science Society would love to host you when you decide to come.
Okay, I look
So actually, I wanna, yeah, I would actually, because Dr. Saad Tanwar, who's the president of our society, Khwarzimi Science Society, he has actually given you an invite, and I'm going to display that on our screen. He says, "We would like to invite you to the next international symposium on biomedical materials organized by IRCBM, Comsat University Samba." So he's basically our president, but he has invited you. So this is like an official, sort of like an official invitation to come to Pakistan quickly.
I would love to come.
All right, I think we have another question. Um, all right, so this is a question from Dr. Mohammed Mustafa. He's asking, what is your most disappointing time in your life? This is to motivate the lady. So, what was the most down, like the lowest thing lowest time you've had, and then how did you come back up?
I can just say to the ladies, life is not easy. When you are a young woman starting out in school, you imagine life to be a different way than it sometimes turns out. I think the hardest thing for me, life goes like this. It doesn't go like this. It doesn't go like that. It doesn't go like this. It goes like this. Okay? So there are peaks and valleys in your life, and you just have to be strong and navigate them. And one of the lowest points in my life was when my father passed away. And I'm a cardiologist and I'm very well trained. And just 2 weeks before his death, I had run a battery of tests on his heart and he was fine. There was nothing, that needed to be done. He seemed to be in very decent shape. And yet 2 weeks later, he died. I was just with him the night before, and I was visiting my parents, and he dropped me at the train station himself on a Sunday night to come back to my job in New York. And on Monday, and I spoke to him on the phone Monday at 1 o'clock. Every day I would have an hour-long conversation with him from my office with my feet on my desk because he used to get up very early in the morning at 4 in the morning. He used to go to bed by 9. So by the time I got home from work, he would be too tired to have a quality conversation. So in the middle of the day, we would have our father-daughter chat. Um, and I spoke to him at 1 o'clock in the afternoon with my usual daily chat. And by 7 p.m., he was gone, sudden cardiac arrest. In the home, he was getting ready to pray his Maghrib prayers and looking at the newspaper to look at the time of sunset and just passed down on the newspaper and was gone. So I mean, like I said, he was everything. He formed everything I am. So that was probably the very lowest time of my life. And things like your mothers, like I say, they pressure you by the time you're 21 or 22, get married. That is the worst advice a mother can give her daughter. I mean, make yourself what you want to make yourself, and then let it happen naturally when it's meant to happen. Don't force it. I mean, my mother tried to force it down my throat. It didn't work out. I just let go of that within 2 months. And I felt like a big burden was released from my shoulders. Um, and I just feel that, you cannot be pushed into these things. You have to really be happy about the person. And for the ladies, I would say it's very important who you marry if you want to have a career. You have to find somebody who's going to be supportive. And it's very hard to do in our culture. So be very, very careful with that. If you're truly ambitious, once you get married, if that person is not supportive, your career is not going to flourish. So that has a very big impact on a woman's career. And for instance, I always say that in the United States, what I see as model communities with this, honestly, are the Jewish community and the Hindu community. They value highly educated and ambitious women. Our culture does not. I'm hoping that things are starting to change, but if you look in the Jewish community, they really value marrying, the men value marrying a woman who is highly educated and also brings an income to the household. Same as with the Hindu community. And amongst the Pakistani diaspora in the United States, I have not seen that as much, although I think that things are starting to change a bit. And so it's very important who you marry, you know.
Thank you so much, and that is such a brilliant advice. And I hope that everyone who's listening and who is actually really ambitious for their career, they do take this advice because it's a golden advice. So I think it's we are almost up the mark of 45 minutes. Time truly does fly when you're having a very good time, and I've had such an amazing time in conversation with you. Thank you so much for taking out the time to join us for this webinar series. Thank you so much for not only exciting and fascinating us about the realm of regenerative medicine, but for also sharing your inspiring life story. Because I know for sure that that story, your struggles really have resonated with a lot of us and they truly have motivated us. So, before I wrap up, is there any message or anything you would like to share? So, before I wrap up, okay, so before I wrap up, one question. What would you say about the Khwarzimi Science Society, a bit about it, and how do you think such societies, how do they play a role in the scientific community, like the work we do
Well,
or we try to do?
I'm very happy to see the existence of this society, and I'm really glad that you're running it with so much energy and enthusiasm. I think in any sort of in a developing country, especially, it's so important to have these societies to encourage the further study of science, to encourage people to go and seek education and training outside the country and bring it back through teaching efforts and training efforts. And I think that I'm just really happy that the society exists. I've never heard of something like this before, and I'm really thrilled that you're running this and encouraging the study of science amongst women, because women, my entire laboratory right now, for the first time, is all women, and I'm so happy about it. And they're brilliant and hardworking, and they're doing amazing things. So women are just in a country like Pakistan. I feel like 50% of the population has just been ignored, and the mind power and the potential, it has to be tapped. So congratulations on this society.
Thank you so much, and it's been a true honor for me as well as Khwarizmi Science Society to host you tonight. And I hope that for any other webinar series, we get to pick your brain again. Um,
Anytime.
Thank you so much once again. Yeah,
Thank you.
and thank you to the audience that in. I mean, the questions that we had today were really good, and thank you so much to the audience for joining in. So, inshallah, take care of yourself, everyone. Inshallah, next time we will be back with another extraordinary Muslim woman in science. Till then, Allah Hafiz. Thank you so much, Dr. Hina.
Thank you. Take care.